Hello, everybody. This is Lab 16, differential media for enterics. There's quite a bit to go over in this lab, a lot of results, um, a lot of tests, a lot of things happening. So make sure this lab especially is a lab that you take some time to understand. Make sure you read the manual, um, look back once you see the pictures of results. I'm going to leave the pictures paused for you guys instead of just explaining everything again, because I'll probably explain it 2 or 3 times before the pictures. So I'll give you some time to pause when that comes up. And if you have any questions, feel free to reach out. This lab especially, like I said, there are a lot of results and a lot of different things. What is the purpose of today's lab? We're going to demonstrate the use of a selective and differential medium for the isolation of gram-negative organisms. We've worked with different types of media in the past that were either selective or differential for different types of organisms, but I think this is the first one that we use that is both selective and differential. So, and you'll see that it's also for the isolation of gram-negative organisms, whereas our CNA plate, think back to the CNA plate, the blood agar plate with colistin and niacylic acid, that was specifically for the isolation of gram-positive organisms. So now we have a way to isolate gram-positive organisms. That's the CNA blood agar plate. And now, after this lecture, you guys will have a way to isolate gram-negative organisms. Another purpose is to learn the basic laboratory tests useful in the differentiation and identification of gram-negative enteric bacteria. You're also going to learn to distinguish lactose fermenting organisms from non-lactose fermenting organisms. We also do that using the selective and differential media that we know as MacConkey agar. So MacConkey agar contains lactose. It's a carbohydrate that the agar itself has, and it has a pH indicator that goes by the name of neutral red. Neutral red has a pH range of 6.8 pH, which is red, to 8.4, which is yellow. And what neutral red does, why it's important, is that it's in this agar because it helps us differentiate bacteria based on their ability to ferment lactose. So does the bacteria that we plate ferment lactose or not? That's one aspect of MacConkey agar. So can it ferment lactose or not? That is one aspect of MacConkey agar. Organisms that can ferment lactose will grow pink to red colored colonies on the MacConkey agar. This is because of there being a decrease in the pH. The environment becomes more acidic. Once lactose is being broken down, it's creating these different acids that decrease the pH. And organisms that cannot ferment lactose will appear clear or colorless on that media. And what they do is they take on the appearance of a non-inoculated MacConkey agar plate. So if you were to not even inoculate the plate at all, that is what this would look like with the organism that cannot ferment lactose. Gram-positive organisms are also inhibited to grow on MacConkey agar by the addition of biosalts and crystal violet in the medium. So there's the other part of MacConkey agar that makes it special, right? We said it's selective and differential. So it only allows things to grow. Well, sorry, no matter what, if something ferments lactose or not, it'll grow. But we'll be able to tell, does it ferment lactose, does it not, based on our neutral red pH indicator. But we also know that we will not see any growth from gram-positive organisms. So the only thing that can grow on MacConkey agar is a gram-negative organism that either does ferment lactose or does not ferment lactose. Okay? So this next slide is we're going to start getting into something called IMVIC, IMVIC. And what IMVIC is, it's a set of reactions that we can use. And what it stands for, it uses the first letter of all these different tests. It stands for indole, methyl red, Voges prosecure, and citrate. So those different tests all make up the IMVIC name. And why do we do IMVIC? It is useful in distinguishing members of Enterobacteriaceae. So using all these tests, these different IMVIC tests, we can perform a test on something, and based on the result, we can say, is it this type of Enterobacteriaceae or is it a different type? So let's talk about indole to start. Indole is formed when the amino acid tryptophan is metabolized in the cell by bacteria that are able to produce the enzyme tryptophanase. So tryptophan is an amino acid. It's formed when bacteria producing the enzyme tryptophanase are breaking it down and forming indole. After incubation, we would have added something called Kovacs reagent. And what Kovacs reagent does is we use it to detect the presence of indole. Kovacs reagent is just a solution of para-dimethylamino-benzaldehyde in an acidic solution of amyl alcohol. The color of this reagent, Kovacs reagent, is yellow to start, but when the reagent reacts with indole, a red color is produced. So if after all of this we add Kovacs reagent to our tube and red is produced, that means that tryptophan was broken down into indole using this enzyme tryptophanase. So what indole is testing for is the presence or the absence of this enzyme tryptophanase. And this slide right here is just showing you tryptophan going to indole plus NH3 plus pyruvic acid because of tryptophanase. So if we add Kovacs reagent and our tube turns red, that means indole is produced, meaning that their enzyme is present. That made tryptophan yield indole and the other stuff, okay? Alrighty. So methyl red, let's talk about methyl red now. Methyl red detects the production of acids formed during metabolism using something called the mixed acid fermentation pathway. And what the mixed acid fermentation pathway is that it uses pyruvate as a substrate to produce things like acetic, lactic, succinic, and formic acids. So mixed acid fermentation pathway is just using pyruvate to create a bunch of different acid types. Methyl red is a pH indicator that is red at a low pH. So when the pH is 4.4 and lower, it is red. And methyl red is yellow at a higher pH. So things that are 6.2 and above will be yellow. Facultative anaerobic gram-negative rods that do not use the mixed acid fermentation pathway usually ferment pyruvate using the 2,3-butane diol pathway. And we'll get into that a little bit more, but first I want to just touch on this picture here. You can see at the very top left, glucose 6-phosphate is going down all the way to 2-pyruvate. That's like the end of glycolysis, right? And it's creating lactate, formate, succinate, ethanol, acetate, and then you see there's some hydrogen and carbon dioxide. So this is the mixed acid fermentation pathway. The pyruvate acts as the substrate, and it is yielding all these different types of acids. So acetate, ethanol, formate, and lactate. Excuse me. So now let's talk a little bit about the next test, Voges prosecure or Voges prosecure, however you want to pronounce it. You can also just abbreviate it as VP. So most members of Enterobacteriaceae use the mixed acid fermentation pathway, which we just talked about. It's using pyruvate to go to those different types of acids. However, there are a couple of species that do not. One of them being Enterobacter, and then some members of Klebsiella also do not use the mixed acid fermentation pathway. So what these organisms do, instead of the mixed acid fermentation pathway, they ferment pyruvate using the 2,3-butane-diol pathway, which we mentioned a little bit just a few minutes ago. So an intermediate in this pathway, the 2,3-butane-diol pathway, is acetylmethylcarbinol, or acetoin. Kind of a fun word, acetoin, not acetone, but toin. And what the Voges Proskure test detects is the acetoin. In an alkaline medium, the acetoin is oxidized to diacetyl, and diacetyl reacts with the creatine in the test reagents to produce a pinkish red compound. So this is all taking place in a basic medium. Alkaline means basic, right? So it's taking place in a basic medium. The acetoin is being oxidized to diacetyl. The diacetyl reacts with creatine. That's in the test reagents, and it produces a pinkish red compound. This test is also sometimes used to distinguish between staphylococci and micrococci. And this next slide here is the 2,3-butane-diol pathway. You can see there, step 4, I've put a box around it in red. That's acetoin right there. So it goes from pyruvate at the top. You'll see that lactate's also over there, acetate, ethanol. So instead of doing just that, which is the mixed acid fermentation pathway, it's going to take acetoin, and it's going to become oxidized in the presence of air and alkaline media. And then you're going to get diacetyl plus creatine from the test compounds, and that's going to give you a pinkish red. That's going to give you a pinkish red compound to end the test with. Okay. So now let's talk about citrate. Some bacteria are able to use citric acid as their sole source of carbon. And there's this type of agar called Simmons citrate agar. And what it has is it contains only citrate as the carbon source. So bacteria can only break down citrate as their source of carbon. And what it also contains is ammonium ions as the only nitrogen source. So using the pH indicator bromothymol blue, we can see our test tube kind of go from green at a neutral pH, turn Prussian blue when the pH rises, and a positive test would be indicated. So the only bacteria that are able to utilize citrate and also ammonium grow well on Simmons citrate agar. If a bacteria does not utilize citrate well as the carbon source, and they can't use ammonium ions, they're not going to grow well, if even at all, on the Simmons citrate agar. Okay? So only bacteria that use citrate and also ammonium grow well. The endpoint is alkaline or basic. We are going to see an increase in pH. Bromothymol blue is going to show us, if it's a positive test, it's going to go from green to blue. And what this means, why this is happening, is because ammonium phosphate is being converted to ammonia. And this right here is what causes our endpoint to become alkaline or basic. And a positive result will be indicated by the development of that Prussian blue color on the slant of the tube. All right. So the last thing we're going to talk about before we start getting to results and things like that is urea hydrolysis. Okay? Urea hydrolysis, the hydrolysis of urea into ammonia and carbon dioxide by the enzyme urease. So one more time, there's a lot of underlines there, but they're all important. Urea hydrolysis is the hydrolysis of urea into ammonia, which is NH3, and carbon dioxide, CO2, by the enzyme urease. And why we care about urea hydrolysis is because it can be used to distinguish different members of the Enterobacteriaceae group. So we know that the genus Proteus and Klebsiella are both positive for urease production, meaning they have that urease enzyme. They can break down urea into NH3 and CO2, ammonia and carbon dioxide. However, we also know that Escherichia and Serratia are both negative for urease production. They are not able to break down urea into NH3, ammonia, and CO2, carbon dioxide. So, the ammonia and carbon dioxide produced by urease, breaking down urea, combine with the water in the medium to give us something called ammonium carbonate. And what ammonium carbonate does is that it raises the pH. It makes it a more basic solution, a more alkaline solution. And how we can tell this is happening, urea agar contains something called phenol red. And what phenol red is it is an indicator that is yellow at a pH of 6.8 and lower. And it is red at a pH when a pH value exceeds 8.4. If an organism produces urease, the slant of the medium will turn magenta. Okay? So it'll turn magenta because it's becoming more basic, more alkaline, and that phenol red pH indicator turns red. So now let's talk about the results a little bit. First is the McConkey agar. I'm not going to talk too much about results. It's kind of like beating a dead horse. You guys have your lab manual, and you can just rewind this if you need to. I just went through and said all the different types of results you might see, what they mean. So if you can't remember, go ahead and just rewind it. It's in the PowerPoint. It's in what I say. So I'll just say a few things looking at these. So on the McConkey agar, some lactose fermenters use pathways that generate more acids than others. And you can see that. You can see the evidence of that in two of these positive results and the other two positives. So you can see a negative result. We know it's going to look just like the uninoculated plate. And a positive result was it's going to grow in that kind of pinkish red colony, right? So you can see a negative result. You can see a positive with high acid on the left here. You can also see a positive lower acid in this middle picture. And then on the right there, it's comparing and contrasting both the positive lower acid and the positive high acid. They're both positive. They're just a little bit different because, like I said, some lactose fermenters use pathways that generate more acid than others. So you can have kind of like a high and low positive. Next is the indole production. We talked about indole and what that means. If you look on the left there, you'll see it's a negative result. And on the right, you see a positive result because of that red, right? It went from yellow to red, which is indicative of indole being produced. And our tryptophanase enzyme is present. Next is the methyl red test. If you look on the left there, you'll see it's negative. It's a negative result. And then on the right is positive. And that is because the methyl red indicator caught that the pH was changing, right? Go back and take a look at that one if you need to. It's on page 66, the methyl red test. Make sure that you're clear what a positive and negative looks like and why it happened, okay? Next, we have that Voges-Proskauer test that we talked about. That is detecting acetoin in the media. And if it does detect acetoin, it turns that pinkish red color, which you see on the right in our positive Voges-Proskauer test. And then the left would be a negative test, nothing happens. No pH change occurred, and our pH indicator didn't catch anything. Then we have the citrate use test. We already talked about that Prussian blue on the slant. You can see that on the right. It's very much a positive result. That whole slant is looking very nicely blue. Unless you're colorblind like me, it kind of looks purplish green. um, yeah. And then on the left here, you can tell that it's not really negative. It's all green. So this one is important because the citrate goes from green to Prussian blue, the green on the left being a negative result, the blue on the right being a positive result on that slant. And then we have the urease test. We have 3 different tubes for the urease test. The first tube is the whole thing turned magenta. This means that it's positive and that the organism produced urease enzyme rapidly. It turned the entire tube magenta very quickly. So the organism was able to produce the enzyme urease, break down urea, and it was very quick. The second tube, the one in the middle, has a magenta slant, but the butt or the bottom of the tube is orange. This is not a negative result. It just means that the organism produced urease at a slower rate than in the first tube. So the urease enzyme is present, it's breaking down urea, but it's at a much slower rate. And then this last one here, the third tube, it's all orange, sometimes it's yellow, but that is a negative result, and the organism did not make urease. The organism may have pushed the media toward yellow by fermenting the glucose in the medium. So it's orange, sometimes yellow. If it's yellow, that means the organism may have pushed the media toward yellow by fermenting the glucose that was in the medium. Okay? And so that just kind of means it had nothing else to do because urease wasn't present. That enzyme wasn't present. So the bacteria in that third one just started breaking down some glucose. And then this last slide here are typical MVIC results. It's a pretty big table. You guys have a similar one on page 70 of your lab manual. Pay attention to the species type. I didn't make this table actually, someone sent it to me. But make sure that you write down the correct results based on this table right here. In your lab manual on page 70, you have everything there. You'll just notice that it's not in order. So on my table here, Alcalogenes faecalis is my first one, but on my page 70 in my lab manual, it's not until the third from the bottom. So just be really careful. Don't mix that up. So you'll see E. coli is the 1, 2, 3, fourth one down here, and you can go over and you can see all the results for E. coli was positive for MacConkey, positive for indole, positive for methyl red, and then negative for Voges, Proskauer, citrate, and urease. But on page 70, that's your number 1. So please make sure you don't mix those up. That's really important. Alrighty, that's all I have for this one. We're all set and done for Lab 16. The next lecture I post will be Lab 17, multiple test media. We're going to be talking about just some other tests we can do. And I think that's the last one before your unknowns. Yeah, that's the last one. Okay. So, this will be the last one I post for today. And then your Lab 17 will be later this week. I'll post that. And then along with that post, I will give you guys some more information on your unknowns. I have a very loose idea of how Dr. Baxter wants me to do it, but I'm going to double check with him and make sure that everything is good to go. And I'll either send out an email saying what I'm going to do, or I'll just mention it in the lecture, okay? All right, guys. Hope you're staying safe. Please email me if you have any questions. I'm here to help. Stay safe, and I'll talk to you guys later.